This Week in Virology, the podcast about viruses, the kind that make you sick. From MicrobeTV, this is TWiV, This Week in Virology, episode 1281, recorded on December 18th, 2025. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Today we are recording at the Sixth Ringberg Symposium on Giant Viruses. It's being held in Ringberg Castle in Tegernsee, Germany. And I have with me today three speakers at the meeting from all different places in the world. They're going to tell you all about their work on giant viruses. So starting right to my left from Germany, Susana Coelho. Welcome.
Thank you.
Did I say your name well?
Perfectly.
Okay, I'm sensitive to people mispronouncing names. Next over there from Canada, John Archibald. Welcome to TWiV.
Thank you.
And finally, all the way at the end, from France, Tom Delmont. Welcome.
Thank you.
I have to say, all three, well, maybe two out of the three have been very nervous about doing this. So make them feel at home. And you can laugh and shout stuff out if you want. That my assistant Karen, who was here last time, was really good at getting you to do that. I'm not so good, but you don't have to be silent. It's great. So I want to start by finding out a little bit of your histories, like how you got where we are today, but go back as far as you can. Where are you from, where you got educated, and how you ended up in your current position. Okay, so Susana, let's start with you.
Okay, so I come from Portugal, from Porto. So I did my studies there. Then I did my PhD in the UK. And then postdoc in France. Then I was in France for more than 15 years. And then I moved to Germany about 5 years ago.
So you're, as an undergraduate, what were you studying?
As an undergrad in Portugal. Yeah, I studied biology.
Biology, general biology?
General biology, yeah.
All right, so then for a PhD, what did you work on?
I worked on algae.
Algae?
On seaweeds.
Seaweed, okay. That's cool. And then you're continuing to work on algae to this day. So why did you decide to be a scientist in the first place?
I don't think I had like this kind of person that loved science since they were 12 or something. I think I didn't really know what to do, and I kind of liked biology. So yeah, I think it was kind of by accident. I really started liking it a lot in the end of my undergraduate program, when I bumped into the life cycle of the seaweeds, and I failed my exam, and I was very offended with really annoyed with myself. And I realized that these organisms have these crazy life cycles and no one knew anything about them. So actually then I did my PhD working on these organisms.
So you failed an exam on seaweed.
Yeah. And then I did my scientific career on seaweed.
Do you know why the virus failed the exam?
Say again?
Why did the virus fail the exam?
No.
It's from last night. Does anyone remember?
Yeah, true, yeah.
Because it kept copying from the host. Yes. I like that one very much. Okay. John, tell us about your background.
Right. Well, I'm Canadian through and through.
Which part, east or west?
I was born in Quebec, but I grew up in Nova Scotia primarily. So I've spent my whole academic career in Canada. I did my undergraduate degree in the 90s, mid to late 90s, in biology. And at that point, I was taking a course on genes and genomes and sort of fell in love with the idea that molecules were able to track evolutionary history. So this idea that they store information and that you can use that to answer important questions. And so I went across campus to do my PhD with a guy named Ford Doolittle at Dalhousie University. And I was pretty much off to the races then. I was in a really vibrant lab where people were answering and asking really interesting questions about evolution of various kinds of cells. And I was just smitten, I guess, with that. I finished in 2001 and then went to do a postdoc at the University of British Columbia with someone named Patrick Keeling, who was and is a prominent molecular evolutionist studying all different kinds of organisms, and then was lucky enough to get hired back into my old department as a professor, and so I've been running a lab there since 2003.
So can you look back and see why you decided to go into biology?
Not really. My first passion in life was cycling, actually. I wanted to be a professional cyclist.
How'd that work?
Not so well, actually. I promised my parents that I would give it a shot and that I'd go back to university if that didn't work out. I probably had no intention of keeping that promise, but biology was one of the very few things in grade school, that I was interested in, and so I was just drawn back to studying that. And the more I learned about it, the more I got passionate about it.
So the Doolittle you mentioned,
Yes.
Is this the Doolittle of homology and that sort of thing?
That's a good question. There are two Doolittles.
Yeah, right.
There's Russell Doolittle.
That's what I'm thinking about,
yep.
probably.
And my boss and mentor, Ford Doolittle, they are apparently distantly related. They worked that out. There's an article they wrote together, I think, on that. But yes, they're related. And they both are in the same field.
Russ always used to go on about percent homology. He didn't like that, right?
Right.
Which is correct. It's
correct.
It's not right to say that. Right. That's why I remember him. Okay, Tom, you're trying to get out of the camera range, but you can't move anymore. You're going to be on camera.
I'm staying with you.
Many people are going to see you. Nice thing to get them nervous. So tell us your background. Where are you from originally?
I am from the southwest of France, not too far from the Atlantic and not too far from Spain. It is not the Basque Country, but almost.
La Rochelle?
No, much more south.
Oh,
yeah.
Give me a city.
Yeah, the closest small city is called Pau, P-A-U.
Yeah, I know that one.
So that's close to it, but even more south in the countryside. By the mountains and nothing around almost. So that's where it started.
Yeah.
And then I joined the closest university available, which was Pau. And they were good in microbiology. So there was a lot of opportunities on this front, and I elected to follow those courses. And within it, very similar to John, molecules. Molecular biology was really the one aspect of it I loved the most. And in fact, the title of the paper that I think is the most beautiful is called Molecules as Documents of Evolution or Evolutionary History. Which I think tells it all, and it's very related to what John said. So then I followed up on not much wet lab, but more of analyzing DNA, from it. So then I moved to the second big city of France, or third, because there's people from Marseille, they will not like it if I say second, in Lyon. So I did my PhD on soil metagenomics with Tim Vogel and Pascal Simonet, who were fantastic mentors. And then I spent 5 years in the US, 2 postdocs, one in Cape Cod, the Marine Biological Laboratory, who was very impactful in my journey in science. 2 trips to Antarctica during this time. The first one was 3 months from Chile to Tasmania and spending a lot of time in the Ross Sea, where the sun doesn't move at all for weeks, to study plankton. And then I had an opportunity to follow someone that was creating his lab at UChicago, and I agreed on it. His name is A. Murat Eren, also known by the name of Meren, and he's my dear colleague and friend, and he's also a big part of my journey. So I did this postdoc with him at UChicago at the Department of Gastroenterology, but I was doing marine biology because I was working on the Tara Ocean's metagenomes quietly from this Department of Gastroenterology. Then I went back to France, and after a year, I secured a position as a public servant at the CNRS, and that's what my position now at the Genoscope, which is a national sequencing center for France. So they contribute to the research of many labs, but they also do their own research, and that is what I'm doing.
And why did you do science? Do you know?
Yeah, so not clear, but for sure I love science fiction. I think it's a theme. There is many of us that share this aspect, and then I guess just, you know, you have to make a choice. It's not completely random, but I like biology more than physics and mathematics, and then I liked microbiology, but it was also a bit of a coincidence because the teachers were studying this. And then slowly, but also not make a choice. In France, we are very lucky. Education is almost free. So, you know, if you don't make a choice, you just continue university. And at the end, it worked well for me.
Okay, you got to be on TWiV. Let me
seems.
just check something. This is the time when I forgot if I turned on the recorder or not. Okay. I forget. I just forget. It's on. Okay, let's talk about some science. So Susana, Tom and John are going to talk about mirusviruses. You and I are going to talk about what you do. So why don't you give us an overview of what your lab is interested in?
Oh, we have 1 hour or something.
No, just a few sentences.
Yeah. So actually, yeah. So my lab is interested in sexual reproduction, sex chromosomes. So the virus, where the virus comes from, well, this is because of the reproduction of the algae. There's a link between the reproduction of the algae and this virus that are involved. I mean,
So you're interested in algae.
Yeah, I think I came to the virus world via the alga world. Yeah.
So you work on a brown
Yeah.
algae. So tell us, I don't know what a brown algae is, and most people probably don't. What is it?
Okay, see giant kelps.
Yes,
Yeah,
those
so
big
that's-
sheets in the ocean,
Yeah, the ones that 50 meters, like forests underwater.
and they can be very wide.
They can be very big, very large. They can be very small.
So this
huge,
is salt water.
This is salt water, yeah. So the brown algae are photosynthetic organisms, but they are not plants. They are not animals. So they are really a different group of organisms that evolved independently from animal and plants. So they invented multicellularity independently from plants and animals. And basically, yeah, they have these crazy life cycles, very complex, that no one knew anything about, including me. That's why I failed the exam. And that's, yeah, so we try to understand how they go through their life cycle and how they reproduce. So they have sex chromosomes, they have males and females. So my lab was interested in, and is still interested in, understanding the molecular mechanisms that regulate their complex life cycles, including their reproduction.
So the genus is Ectocarpus, right?
Well, there's Ectocarpus, but there's also these kelps. There's more than 3,000 species. Yeah, many species. So we work with many different species.
So it does photosynthesis, but it doesn't have chlorophyll?
It has chlorophyll also, but then they have other pigments that
Make
give
them brown?
them the brown color. Yeah.
Okay, I was wondering. So they can be very large, but they can also exist
Be
as
tiny,
tiny cells, right? And so are these ever a problem? Do they, single-celled algae can cause blooms, right?
Yeah.
Yeah, but do these ever cause some problem?
Well, they do. The sargassum see the problems with the growth of sargassum blooms. The sargassum are brown algae.
Okay.
But otherwise they are, except that they bother you when you want to go swimming and you have them around. Some people don't like them, but they are good for creams. So actually,
What, hand
you know
cream?
that? Yeah, hand
No, I didn't
creams.
know
You know
that.
that. Yogurts and ice creams and all these things they use. So these organisms have this cell wall that have some specific sugars. So they are very much used for as thickening agents.
John, I went to Vancouver Island once, and the whales, they lick the kelp on the surface of the water to feed on the whatever's on the kelp. Did you know that?
I did not know
It's very
that.
cool.
Yeah.
You go out in these boats, and the whales come up, and they tilt sideways, and they just lick their tubes. These algae are tubes. It's just amazing, right? It's very cool.
Fantastic.
So you say you use this brown algae as a model for evolutionary.
Yeah.
So how do you do that?
We do genomes, so we look at what's in there, what are the genes that are there. Most of the genes have no known function, so we use mutants to understand their function. So for that, we developed this model organism for the brown algae, that's Ectocarpus, and a few other species now. So we can have molecular techniques to understand the function of their genes.
Right. So tell us the reproductive cycle. How does it work?
I need a drawing. I need to draw it. How can I explain it? So they have males and females, but the males and the females are haploid.
And they're just
diploid.
single cells, right?
No, they are multicellular.
Okay.
So they are, in the life cycle of the algae, what you see, so the giant kelp, you see the sporophyte generation, so the diploid stage. But then there's meiosis in this diploid stage, and then they produce males and females that also grow, and they are multicellular, but they are completely different morphologically from the diploid. And they live elsewhere, actually, in the sea. So actually, before, people thought they were different species, actually. So you have very tight regulation of developmental program, depending on whether you're in the haploid or in the diploid stage. So we studied these genes, the genes that trigger the male versus female development, so the sex chromosomes, so they have specific type of.
So can you do this in the lab, in a tank?
We can do that in the lab, yeah. So no swimming pool.
So you can.
So it's very very boring actually. We can grow them in petri dishes.
Do they? Petri dishes.
Yeah,
No
yeah,
tanks.
Yeah. No, the kelps we grow them in 1 liter flasks.
Yeah.
And then we can manipulate the light to induce meiosis, so we can do the full life cycle in the lab.
Okay.
So, yeah.
So you said you do genome sequences, right? To try and understand various questions, and that's how you became an accidental virologist, right?
Yes, kind of. Yeah, exactly. So when we sequenced all these genomes, we realized that there are a lot of these endogenous viral elements, so these potential proviruses, and also a lot of bits and pieces of potentially virus that are now host viruses, but probably with a viral origin. So we start getting interested in.
So if you do a genome sequence, how do you know if something is an integrated viral genome?
Because you do blasts and you find that there is this this old piece that corresponds. Well, to be frank, the virus that infects Ectocarpus has been known for already a long time, since the 80s. But people didn't really know the details of the integration sites and all that. So, but it was already known for many years that there was a virus, a giant virus that was having a coordinated life cycle with the host and the infectious cycle of the virus. So this was already kind of known, but not in a lot of detail.
So can you take this virus and add it to your petri dishes and infect cells?
Not really. So what we have is we have strains that are viral producers. So we can then cultivate these strains and then make them release their virus and infect other strains.
Okay, so that released virus then you can put on another and it will infect.
Yes.
Okay. But you first saw it as a integrated DNA into the algal genome, right?
Yes, that's. I knew already that there was this Dieter Müller's lab that had studied this virus for a long time. So I knew that existed. But when we saw in these genomes this amazing quantity of inserted elements, then that started being really exciting.
So is it correct that the virus only reproduces in the gametes?
Yeah. So contrary to most of these giant viruses that are, you guys work on unicellular organisms, so the brown algae are multicellular. So they have different cell types, different organs, and so on. So these viruses, they are only lytic in very specific cell types. So in the gametangia, so in the flowers, if you want, of the alga. So they are silenced. So there's no symptoms in the vegetative tissue. And they only replicate in these specific structures.
When they do replicate, do they destroy the cells?
they are very well coordinated with neighboring cells that release gametes. So at the moment where there's gamete release, actually this structure opens and releases viral particles instead of releasing the gametes. So it reduces the fitness, if you want, of the algae. It doesn't kill it.
It's a coordinated event.
Yes.
That's very interesting.
Yeah.
To find out how that works.
Exactly. So
right?
this
is that one of the things
that
you're
we are
interested
really
in?
interested in. Yeah.
So does every brown algae have this virus integrated into it?
We don't know exactly. So people have been looking by PCR, and they think that there's at least 80% of the collected algae have. But it doesn't mean that all of them have active or potentially active inserted.
So you think this integrated DNA, by the way, you call it an EVE, right?
Endogenous, yeah.
Endogenous viral element. But you also use the word provirus, okay? You better be careful with provirus because that has a very specific definition.
I guess it's one that is capable of...
Yes. And I mean, it was first defined with retroviruses, right? Howard Temin. Anyone know Howard Temin? Remember that name? You better. He was the first to propose the provirus hypothesis of how RNA tumor viruses transform cells. They make a DNA copy and then it integrates. And that led to the discovery of reverse transcriptase. So provirus was coined as the name of the integrated retroviral genome that gives rise to new virus.
Okay, so this is the way I'm using it.
So if yours gives rise to virus, then you could call it provirus. But
so on top of that, there are a lot of endogenous viral elements
that are not complete genomes. But the one that's complete, you're sure that gives rise to virus particles, right?
Yes, so this is what we've been studying using genetics and to associate the provirus with the symptoms and the virus that is released in this structure.
So you think that the provirus is an obligatory part of the reproduction cycle?
No. It is. It replaces the gametes in these structures because it gets active. It is activated in these structures, and then it replicates and it, let's say, sterilizes the algae. But it's not necessary because you have by meiosis you can get rid of this provirus because it is inherited as a supergene, right? So half of the daughter cells will not have this integrated provirus if you started, of course, from an heterozygous alga. So it's not necessary for the reproduction. But it's coordinated with the reproduction.
So you could take virus particles and infect cells, and it would reproduce without integrating.
The alga?
No, the virus.
Or the virus. We don't know, but possibly.
That's what I mean. Is it obligatory? So for retroviruses, it's obligatory. You cannot make a retrovirus without a provirus. You're not sure?
It's not, no. I don't think it is.
That's an interesting question, right?
No. Well, the only thing I can say is that we have infected the gametes, because I didn't say that, but the virus only enters the gametes because the gametes don't have a cell wall. And the only thing we know is that we can infect gametes, and then we can see that there are, by PCR, we can see that the virus is in the cells, but we don't know if it's integrated or not. I don't think it's integrated because in the next generation, by PCR, we don't detect it anymore. So it's possible that integrations are not always successful. So in that case, it stays as an episome, I guess. What we were looking at was with the PCR, it was just the episome, the viral episome.
So do you have any idea of how the virus gets into the cell, into the gamete?
There are some photos from the 80s with the TEM where you see the virus like at the surface of the gametes and then entering, but nothing is really known in detail.
Is this virus, what does a virus look like, by the way? It's a big virus, right? Icosahedral?
Yeah.
Does it have a membrane?
We don't know.
Don't know. But it's
not
just,
me that doesn't know. No.
Yeah, it's just spherical, right? It's a spherical particle.
Inside, yes. I never know how to do it.
Icosahedral,
yes, and then it's, yes.
But you could easily find out if it has a membrane,
yes.
right? It's an easy experiment to do.
Yeah, we'll know more.
So, I read in one of your papers that virus production is induced by low temperature.
Yes.
So in the ocean, isn't the temperature always the same?
Good question. So these algae are in intertidal zone, so the changes in temperature are more fluctuating. So it's not like open ocean.
Are they surface growers mainly?
Yes, they are in the tidal pools, tide pools. Yes, so the virus is only active, so the symptoms are visible at 10 degrees. So if the algae is at 14 or 20, then we don't see the symptoms. It's only at 10 degrees. But I think this is part of the coordination between the algae and the virus. This is how probably it's been maintained for so long, because the virus doesn't kill the organism, right? It just sterilizes it slightly, but then if the temperature is 14 degrees, then it doesn't sterilize it. So there is this very nice interaction between both.
Can you reproduce this temperature effect in the lab in your dishes? Yeah. That's how you get virus.
Exactly. Yeah.
Now, if you infect fresh cells, do you have to keep them at a low temperature?
Yes, if we want to see the symptoms, yes.
If you want to see the symptoms.
Yes.
So, but what about producing new virus particles?
I think it doesn't happen at 14 or 20 degrees.
Interesting. Are you interested in knowing why?
No.
No.
We don't know anything.
No, it would be interesting to find out what is the temperature sensor that triggers the virus to reproduce.
I don't know.
So let me get something else straight. First, so there's an integrated copy, which may be a way of storing the genome for the future. There's also an episomal copy?
Possibly, yeah.
Possibly? I thought in your talk you said there's an episome.
No, what I said is that the circular, it's the way the virus gets activated via that phase in the replication where there's a circular. That's what I said. And then I said that possibly, but I didn't talk about that in my talk, possibly there is also episomes, because we see by PCR that the virus is there, but it's not integrated. That's for sure.
So in the virus particle, the genome is a circle, or is it linear?
I don't know.
Don't know?
No.
That's easy enough to do also, right?
Yeah.
You can cut it or you can sequence it, I guess. But, right? Couldn't you do that?
Yeah, I'm sure we could, yeah.
Yes.
Just ask
That's
them
in my
how to
list.
do it.
I'll put it on my list, to-do list.
So if someone came up to you, not a scientist, and said, "Why are you studying brown algae?" What would you tell them?
Because no one studies them. They are fascinating, and they are creative organisms. They just reinvented a lot of. They look like plants, but they're not plants. They reinvented everything. Yeah.
And sometimes they cause problems, right?
Yeah, but that doesn't.
It's not nothing.
Nah, that's okay.
Okay. Are they endangered?
Yeah, beautiful. Well, they are going north because they don't like hot water.
Yeah.
So the populations are kind of moving north. Some populations are changing, not endangered, I would say.
So, right. This virus, what do you call it? Brown algae virus? What's the name of it?
Ectocarpus siliculosus virus.
Okay.
Yeah. This is the classical name.
And as you said, there are a lot of kinds of brown algae. Do they each have their own viruses?
Yes, apparently. So this work that was done in the 80s showed that there are at least some like 3 or 4 species. There's specific virus that infect those. And actually, the virus of Ectocarpus, for example, will only infect Ectocarpus species, not other closely related species. So there seems to be a species specific association between virus and it does not infect. As far as we know, spores, so it's very specific for the gametes. So we think there is maybe some receptor on the gametes that allows the virus to infect them.
Okay. So one thing I noticed by coming to these meetings 4 times is that people study algae and single-cell eukaryotes, and then they stumble into a virus, and it grabs them because they can't resist. It's so interesting, right? And then they become virologists. I think this is really interesting. In the old days, you just became a virologist. You didn't stumble into it. But and that's so everyone says, "Well, I'm not a virologist, but here we found this virus and we have to study." That's very cool. Okay, now Tom and John, you both work on. Is it mirus or mirusviruses?
The way I say it is mirusviruses. Mirusviruses.
Mirus, but virus it's like virus with an M, mirus, right? So
Maybe I said it wrong, but
No. I'm just asking. So where did the name come from to begin with? Mirusvirus.
It came from the fact that there was a need to name it, and then there was, a struggle to find a good name to not have any regret. And Mirus has the advantage of being short, and it's from ancient lineage, ancient language to say something unusual or strange, and there is a few other words for it, but yeah.
But Mirus. I'm going to say it.
It's okay.
You didn't just take virus and change the first letter, did you?
Say again?
You didn't just take virus and make it.
Oh, no, not at all.
Okay.
No.
Mirus.
No, I looked for anything from Greek or Latin that will look okay in light of the very little knowledge we had of those viruses. We still have little knowledge, but now we have more.
So your paper, Mirusviruses link herpes viruses to giant viruses. We did that on TWiV 1005. So it was about 250 episodes ago or more. So I want you to just summarize that for the listeners, because it's a long time ago. So we're just briefly.
Oh yes, because I
forgot to tell you. I forgot to ask you. Tell us the 2 sentences. What's the interest in your lab? The general interest. What are you interested in?
Oh, in my lab,
yeah.
I study plankton. So my main interest is to understand plankton from the light of genomics. So I've been doing bacteria, archaea a little bit, not much, eukaryotes. So then I was getting closer to the mirusviruses, unicellular eukaryotes. And then, very similar to what you said of many people, I just, in a way, didn't meant to be a virologist, which I am not, but I am indeed dedicating a lot of time on those viruses now. But really much from a bioinformatics perspective, sadly, because when I see all of those amazing sessions and those images and now videos of particles and cultures and control experiments, I'm very far from it. So no wet lab, and a lot was done from the metagenomic legacy of Tara oceans. So maybe later we will go into the detail of metagenomics, but so the broad interest is I come more from the ecology side of plankton, but because of those viruses, I had to learn a lot about evolution. So now I enjoy almost those phylogenies and and taxonomic rules and names and so on. So yes, so that's
okay. So this paper that I mentioned, the Mirusviruses and herpes viruses. Just briefly summarize that. What happened? How did you stumble on these viruses?
How I stumbled is not short. Maybe I start on what they are, or is it okay?
Say whatever you want.
Yeah,
sure.
Yeah. So those are double-stranded DNA viruses. So that's for sure. They, on average, they will have 250 kilobase of genomic size, circular. So maybe 250 genes, more or less. And it seems that they infect a broad range of lineages of unicellular eukaryotes, so protists, which happen to be very abundant and diverse at the surface of the oceans. So because the food supply, the eukaryotes are very abundant, then the viruses are also very abundant. And so when the Tara expedition did what they did, which was to sample a lot of different size fractions and depths at many locations at the surface of 2 seas and all the oceans, there was a lot of signal for those viruses. But because they are very divergent in terms of evolution from what was known at the time, it was not that easy to, understand that those were viral genomes and to understand what is their evolutionary history or functioning. So that's why there was some time before those viruses, even though they are quite abundant, in many aquatic ecosystems especially, were not identified until 2023. So, they infect a lot of unicellular eukaryotes. So of course they play an important role on the ecology of plankton, because they contribute to lysing the cells, we assume. And right, and in terms of evolution, there is also something particular, and that's related to the title of the paper. What happens is most people in this room that are dedicating to studying, especially not only, but a large group of viruses, which are called the nucleocytoviruses, which is, I think, also related to what Susana was mentioning on her model. So those are very, very abundant and diversified, and people have been studying them for 2 decades or more. But those have a particular type that is completely unrelated from another well-known lineage of eukaryotic viruses, which is herpes. And so the nucleocytoviruses have one type of major capsid protein, which is a rule of taxonomy those days. So they are in one basket of taxonomy. And herpes viruses, those infecting animals, have another type of major capsid protein, so they are related to another realm of viruses. So those are evolutionarily disconnected, at least from that perspective. And the viruses we found had a lot of functions similar to those of nucleocytoviruses, yet they had a particular type of herpes viruses. So that's the connection. In terms of evolution, they are on the side of herpes, but in terms of functioning, there is less separation between herpes and nucleocytoviruses because of the mirus. Does it make sense?
Yeah, I got it. So you say that these mirusviruses infect many organisms in the ocean. How do you know that?
Yeah, that's why I say...
You say you don't do experiments. You don't do wet experiments.
Exactly. So first, I'm very happy to see that multiple groups of people have been studying them since 2023. So all the insights John and I will present are not only from our work, but also from other people's work. But from multiple ways of predicting hosts, multiple groups of people see the same thing, which is that they infect a broad range of unicellular eukaryotes. For example, if you look at the genes in all the legacy of culture eukaryote genomes, now that we have the genomes of mirusviruses, and especially the key marker genes that are very important for especially making the particle, you can search for them, and you realize that many of those genomes in cultures that have been sequenced for decades, they do contain specific genes related to mirus. So that's one hint, okay? Now, in addition, we also have a lot of environmental genomes of eukaryotes that are also coexisting in the surface of the ocean, for example, and they do share a lot of genes that suggest maybe gene flux, and that could, that are, again, other hints to suggest what are the eukaryotic lineages that they primarily infect. So none of this is demonstrated, but that's where at some point we'll also hear from John, who has some hard evidence about the host of some of those at least.
So the mirusviruses, do they only infect single-celled?
Yeah, it's a good question. I will say at this point there is, and if John has something to say on this, you can jump in anytime, by the way. So far, there is no clear evidence in my view. So sometimes we detect signal in coral, but it does not mean they infect coral. They could infect unicellular eukaryotes inside the coral.
Yeah.
So at this point, that's, no clear evidence they infect multicellular organisms.
And so how many different mirusviruses are there?
Yeah, it's very, very diversified.
Is it the most diverse of any virus?
No, I don't think so. I will say, again, I am new to this field. My sense is people doing phage, infecting mostly bacteria. So there is this class called Codoviricites, tailed phages. It's possibly the one that is extremely diversified.
But
so
this is up there. It's high, right?
Yeah, but the thing is, mirus is related to those as well. I didn't go into that part, but mirus fits a gap between, or maybe I said it, between tailed phages and herpes. Because the three groups have the same particle type.
So this means they have a common ancestor?
So it means it looks like it.
Yeah, it looks like. Right.
It's difficult to demonstrate it. Some people argued this week that they could be, parallel convergent evolution to reach the same particle type, but at least in my mind, it's more likely that it's a continuous evolution.
So mirusviruses don't infect brown algae, right?
As far as I know.
So that's the thing. They are so divergent in terms of genomics that the community has to adapt and to also use those databases to search and detect them. So if you don't use the genomes to search for them in your data, you will likely not see them. So
Yes,
possibly.
Yeah.
Okay, so now, John, you also stumbled onto a virus, and it turned out to be a mirusvirus, right? Can you tell us that
Indeed,
story?
yeah, and what you said earlier about people just doing their thing and realizing, my lab we're very much studying biology and evolution through the lens of comparative genomics, so we are good at staring at DNA sequences and making inferences about events that have happened. And one of the things that we've studied over the years is organelles. So these are things, mainly the mitochondria of eukaryotic cells and the chloroplasts of plants and algae. And they have separate origins from the eukaryotic host component. So we can see that the word I usually use is mosaicism, so genome mosaicism. So using bioinformatic techniques, you can look at genomes and determine that different genes within the genome have different evolutionary histories. And so we never really set out to study viruses in particular, but really what's transpired over the past decade or so is that you can't not be interested and pay attention to viruses if you're studying nuclear genome evolution, so in particular in microbial eukaryotes. So we literally stumbled on the mirusviruses. By the way, I say mirusviruses sometimes too. I usually say mirusviruses just because Tom does, but I'm comfortable with either. We were helping colleagues in the U.S. at Stony Brook, two scientists, Jackie Collier and Josh Rest. We were helping them assemble the genome of a particular single-celled eukaryotic organism called Aurantiochytrium. And by the way, it's actually related to brown algae and kelp, so shares a common ancestor with those multicellular forms, but it's a single-celled organism. And we were using a particular technology called nanopore sequencing, which is a fancy new tool that allows us to get the individual DNA sequencing reads much longer than were previously available. So this technology has really transformed genome sequencing and assembly. And long story short...
We're talking about kilobases of sequencing.
Yes, kilobases, 10, 20, 100, 500 kilobases in an individual DNA sequence read.
And the error rate is not bad.
The error rate has gotten better and better over the years. Initially it was a bit rough and ready, but it's improved dramatically. So we were helping Jackie and Josh in Stony Brook. That was a genome that was sequenced by the Joint Genome Institute in California originally. And I had a postdoc at the time, Lucie Gallo-Lavallee from France, and she was an expert in detecting viral signatures in genomes. And we were already kind of drifting towards this area, which is one of the reasons Lucie ended up in my group. But long story short, Jackie and Josh had identified a couple of sequences within the Aurantiochytrium genome assembly that were virus-like. And at that time, that was about the most that could be said. Again, we can, with bioinformatics, we can identify these signatures of viruses when you have enough reference information. So there were these two distinct regions. One of them was an integrant, a chromosomal integrant, so one end of chromosome 15, as it turns out, about 300 kilobases, was a region of putative viral origin. And there was also a second element, an episomal element, so a circular molecule that also had viral signatures. And my recollection is we were really struggling at that time to move beyond just saying these are virus-like elements. And then, as luck would have it, there was a big data dump, and it is the paper that you and Tom have just been talking about. So this is Morgan Gaia was the first author, along with Tom and colleagues. And Lucy, my postdoc, as soon as that data were available, I think it was a preprint at the time, she started doing comparative work. She was alerted to it probably automatically that this paper was out. And she said, I know what those are. It was one of these cases where without proper reference information, you can't make sense of these very divergent sequences. And she said she knew what they were, and the more we analyzed, the more we said, those are mirusvirus elements.
When did you call him?
I didn't call him. I don't really remember why, Tom. But we actually, I do know why. We were, I'm pretty sure, hell-bent on getting the paper, which was already, fully drafted and telling a story about this original genome. And then the more we studied it, the more interesting it got. And then the Gaia et al. Paper came out in Nature somewhere along the line in 2023, I guess, maybe early 2023. Mm-hmm. And we, more and more the focus of the paper became that this was a putative host of mirusvirus. Mm-hmm. And so, the organism is called Aurantiochytrium, and we ID'd 2 distinct mirusvirus elements in the genome.
So I understand you can make supplements from this whatever.
Yeah, we'll just call it Aurantio.
Aurantio.
Yeah.
Sure. Yes.
Aurantio.
So algae and these guys are useful. In your case, they're single cells, right?
That's right.
They don't make aggregates at all.
They do a bit of clump. They're a bit clumpy.
Clumpy, okay.
Yeah, but they don't form any, differentiated multicellular forms.
So you saw the DNA in the genome. So where are the viruses?
Yes. Okay. So we got the paper published, the original genome description, but of course sequences are sequences, and we were pretty keen even before that paper was out to actually visualize the viruses. We knew from Jackie and Josh's work on the transcriptome that these viral elements were giving rise to transcripts, so that made sense. And then it was a matter of starting to do experiments to see what we could see. Initially, we did some scanning electron microscopy, if memory serves. Naively thinking that we would just see individual cells that were just decorated with virus particles. That turned out not to be the case. Then we switched over to transmission electron microscopy, and initially the results were not very helpful, but really the champion of that line of inquiry was a postdoc named Dudley Chung, and he looked at enough images and was patient enough to stumble upon sections of through Aurantio, where it was very clear that there were viral particles in the nucleus, in the cytoplasm, sort of on the order of 120 to 140 nanometers across, and they looked not unlike herpes virus. My colleague Craig McCormick runs a virology lab. One of the viruses he studies is herpes viruses, and he's just a floor down from me. We had never collaborated, and so it became a very fruitful collaboration between my team and Jackie and Josh at Stony Brook and the McCormick lab in the same building as me. And they're the card-carrying virologists, and they have a very interesting perspective, and we've learned from them, and they've learned from us.
So I'll ask you the same question as I asked Susana. So is this integrated copy an obligate part of the reproduction cycle?
Short answer is we don't know anything really about reproduction. A moment ago when you asked about whether they're multicellular, and I said they were clumpy, the life cycle of these particular organisms to which Arantio belongs are called thraustocytrids. They were confused. They originally classified as fungi many years ago. They have fungus-like properties. And so they have a very complex life cycle that we're not able to control. We can see gametes, so flagellated zoospores in culture to a low level. They'll also undergo division within a larger cell wall, so you end up with multiple distinct cells within a large bag, if you will. So they're not an easy system.
Can I ask a question? They are haploid or diploid?
Good question. We think haploid, but there is
From the genome,
undoubtedly, well, sometimes you can tell, sometimes you can't, but we would predict that there are both of those stages somewhere embedded in the life cycle. The genomes are relatively easy and straightforward to sequence and understand, but taming the life cycle in a controllable way that we can do experiments to dissect the virology is rather difficult.
So are you able to purify particles in genome sequence and show that it's the same as what is integrated?
Excellent question. Dudley was able to purify virus particles from extracellular material. It was not easy work, but we did that. And they did various molecular techniques. We never did single virus sequencing. We could never get enough material to do whole genome sequencing, but we could use digital droplet PCR, which is a very sensitive technique for quantifying the presence of viral DNA in these viral particles. So we do see the DNA from both of these two forms in virus particles outside the cell. Although I will say the episomal version is the one that seems to be far and away the most abundant in terms of giving rise to virus particles.
That is in the cytoplasm or the nucleus?
Well, presumably it's in the nucleus.
Like herpes DNA,
Yeah.
right? Yeah. But in the virus particle, is it circular?
Oh, well, we don't have no knowledge.
Because herpes is linear, right? It gets in the nucleus and is circularized by ligases.
Yeah,
That would be interesting if it does the same thing.
Yeah, we have no knowledge of that.
But can you take these particles and put them on fresh cells and get an infection?
Okay. The short answer is no. I want to mention again the collaboration with Craig McCormick, because this is interesting where people from different disciplines have to spend some time learning the same language, because both of these elements are baked into the organism. So we have an integrated nuclear copy, which is V2, we call that V2, and the episome is V1, but as far as we can tell, they're both always there. So the concept of an infection experiment is formally possible, but you can't really know without doing manipulation of some kind.
Well, you could remove those, right?
Well, we could, yeah. There is, people are developing genetic tools with the thraustocytrids, and Jackie Collier's team are doing that.
I mean,
so in the fullness of time, we could trash both of them and see what happens.
That would be, and then infect,
Yeah. And we have found, I will say too, a graduate student, Jess Latimer, and some undergrads are looking for new strains in nature, and we have found at least one strain that does not, a strain related to Arantio that has no obvious virus in its genome. So that's potentially a naive host that you could do infection experiments with. That's kind of state of the art right now.
So this is the closest to an infection system for a mirusvirus. Mirusvirus. Yeah. Mirus.
Yeah.
That's the title of this episode, Mirus or Mirus.
Yes. Yeah.
That's the closest we have. No other mirusvirus can infect a cell, right?
Well, that is known. So that's really where my world and Tom's world kind of started to cross-fertilize one another. I think from the Guy et al. Paper and other ones, you can detect and predict what the probable hosts are. And so beyond the little tiny corner of biology that I've been telling you about, Arantio and friends, it appears that there are mirusviruses that are infecting a whole raft of diverse eukaryotes. To my knowledge, the thraustocytrids, to which Arantio belongs, is the only one where the visualization has been done, and we've got experiments. But I will say too, one quick acknowledgement. Jackie Collier discovered a paper published in 1972 by Kazama and Shorenstein, and they described a herpes-like virus in a fungus, quote unquote.
Right.
And that's what these organisms were known as back then, fungi. So the first visualization of what we believe are mirusviruses goes back to the early 70s.
It's interesting. If you, in those days,
Yeah.
that's all you could do is look at cells. But if you never looked at a cell, you would never know there's a virus. And we didn't have genome sequencing, right? But people who study protists, for example, they look at their cells often. And you can discover things by looking at them.
Absolutely. And that's a third hub of this is the protistology community, to which I'm connected, but I'm by no means an expert. That know thy organism, you know.
Well, you have to have someone who's interested too, because like that 72 paper, they see particles and they're not virologists. What are they going to do? It's in the record and it's left for someone else to pick it up.
Correct.
But I'm really struck by, so in early years of virology, so I entered virology in the 70s, but it had already been a 70-year-old field. Most viruses did not integrate as part of their reproduction cycle. They were discovered as exogenous particles, right? Mostly because they cause disease, and then you would isolate the agent and you would have the virus. So this idea, which I've heard over at this meeting, that you look in the genome and find the virus and go from there, that's really contrary to what most virology has already been. It's not bad, I'm not saying that, it's just really different. And it's because you're interested in genomes, right? And we have the capacity to do that. When I worked on poliovirus, I sequenced the genome. It was one of the first RNA virus genomes sequenced. It took me 1 year to do one virus. And the read length was 200. And we had 2x coverage as well. So you could not do more than one. It was really so different. So this really impresses me at being a different way to do virology, kind of from the back, right? You don't start out to be a virologist. So when did you hear about this Orantio, right? Orantio and virus?
Relatively early. But after the paper was published, maybe not immediately, or maybe yes, I don't know exactly, but we had a Zoom call. That's the first time we discussed. Owing to the distance between France and Canada, it was more convenient. And we shared our excitement about those viruses. And I think again, we did not know each other, but at least from my perspective, it was easy to trust John. And we quickly shared unpublished results from both sides without any restraint. And we have done a few others since then and doing the same strategy. So we are quite open between us on what we do and what we learned on those viruses. So when he showed me the first images of the particles, that was not published at the time, because this is the second paper, correct?
Yeah.
I was moved by it, because again, it was all done from a bioinformatics perspective. And even though with Morgan and Mark Kupovic, Morgan Gaia and others, we were quite confident in the reality of those viruses, which were viewed as chimeric between nucleostoviruses and herpes viruses. Some people that don't have the expertise of metagenomics or didn't look at the data, it will be completely normal to not fully embrace it.
Yeah.
And even for me, even though I was very confident, to see those particles was a good moment.
Well, there are people, and you probably have heard this, who say if you don't have an infectious virus, you don't have a virus. Genome is not enough. Now, I know most people here will disagree with that, but all the other virologists out there, right, that they say, you have a sequence, show me the virus. But that's what you're working towards, because you can't really do much unless you have a cell culture system. If you want to understand how the virus works, that's what you need, right? So do you think, these are enveloped, these mirrors,
Yes.
right? Do you think they replicate in the nucleus?
Okay, so this goes to... Okay, maybe it's a time I will tell a bit the story of how we found them and how we found more, if it's okay. So, I don't know where to start. I don't want it to be too long. But I need to mention my colleague Meren, who is a computer scientist, and he really led the development of this bioinformatic platform called NVO, which is being used by many people to do all sorts of analysis on genomics and more, comparative genomics, phylogenomics, but also to as a means to extract genomes from metagenomic assemblies. So, what is a metagenomic assembly? It comes from a metagenome. What is a metagenome? Is when we go and sample the field. So it could be, for example, a liter of water from the ocean, as was done for Tara oceans, and then you will kill everything. And then sequence the DNA deeply now, short read or long read, as you said. And then all of those little fragments are from all sorts of genomes. So as a first very important step, you will do an assembly. You will use software to find short sequences that belong to the same genome, and they will produce much longer sequences, okay, which we call contigs or genomic fragments, if I want to keep it simple.
You know, in 1976, we called them contigs also.
Perfect.
There was a program called Staaten, which maybe some of you remember. They made contigs.
Okay.
Yep.
Perfect. So then you have a certain number of contigs. It could be a few thousand, or it could be a few million. It could be very simple, or it could be very complex. And then there is ways to try and often success in putting together contigs that belong to the same genome. With bioinformatics. So you do what the assembly could not do, because the assembly will not reach to entire genomes often, even though now it's getting better and better with long read. So you will have to put together to solve the puzzle yourself, in a sense. A key aspect of solving the puzzle is if you have many samples and your genome is detected in multiple samples at different level of signal. And the very key thing that works for bacteria and archaea and eukaryotes and viruses is that the fragments of the same genome, they will almost perfectly correlate across samples. And they will not correlate with the other fragments of other genomes, because most often those populations will not perfectly correlate across samples. So it works quite well. There is other tricks to do what we call binning. The official naming is called genome resolved metagenomics. So how do I get genomes from my metagenomes? Now, NVO, developed mainly by Meren, offers means to process your data and to visualize it in order to identify and extract genomes, and also to gain confidence, because you look at the data, you see the signal. For each of your contigs, you know their abundance, you know other aspect of it, and then, for example, for bacteria, there is ways to assess if it's a good-looking genome or not based on the occurrence of single copy core genes. Okay, so it will just count. And if all of them are here, it will be a beautiful genome. But if some of those are detected multiple times, there will be a flag to say, be cautious, maybe it's contaminated. Maybe there is multiple genomes in your, in what you think is an environmental genome. So there is ways to extract them and then assess if they are good or not. Okay. Fine. So this is quite, all of this was developed mainly for bacteria at first and archaea, and then later on was applied to eukaryotes and also the viruses. So we had published the platform NVO in 2015, which is the exact same year of the release of 5 beautiful papers of Tara Ocean, all with the data being made available. And when I say data, for me, the important part is the metagenomic data. So it was a huge volume, really significant volume of metagenomic data dedicated to plankton. So we just published a platform to extract genomes from metagenomes, and then there was this beautiful cake on the table that was not eaten, because at that moment, the consortium of scientists that carried on the project of Tara did not try to do that, because at that moment it was not common to do it. So we took all the data and we started extracting bacterial and archaeal genomes from all of this metagenomic legacy. We got maybe about 1,000 of them, and we, did some science with those genomes. And then I came to the Genoscope in France, where I do my research. And in this institute, which is where all the sequencing of Tara was generated, they are a big part of Tara Oceans. Their main objective is to study the unicellular eukaryotes within the scope of the project. So with them, I started to try to extract genomes of the eukaryotes instead of just the bacteria and the archaea. And to some extent it worked. We got hundreds of environmental genomes for protists. Not all of them, a few of them, but it was already useful. So I was done with the cells. I had a short postdoc at the lab of Patrick Forter, and this is where I met Morgan Gaia, who was a postdoc with him at the time. At that moment, I have not done a single work day on viruses. So this is 2018. And they explained to me what they do. They work on this key marker gene, RNA polymerase, one of the subunits. Why? Because it's a very constrained gene in terms of evolution that is present in all of bacteria, archaea, eukarya, and a lot of the giant viruses. And they use it to try to, among other things, estimate the age of those giant viruses. Mm-hmm. And when I come to them, I have all of those metagenomic assemblies from Tara and my knowledge on how to extract genomes from bacteria, archaea, eukaryotes. And they tell me about this marker gene that I didn't really think of. And I realized we could explore the metagenomic data from the eye of this one marker gene using phylogeny, so using evolution as guidance to go after the giant viruses. So we did that. We found a lot of signal for the giant viruses that were already known. But again, from the perspective of a single marker with a phylogeny. So we could see clades related to Imiter virales. And we could see clades related to Pandora virales, orders of giant viruses. And every tip of the tree is an opportunity to get a genome. So sometimes there is no good-looking genome, sometimes there is a good-looking genome. So we take as much as we can. So again, maybe 1,000 of them. And then there was deep branching clades with no representatives. So we can see a phylogenetic clade, it's very clear. So those are related RNA polymerases, but they are not related to anything known. No viruses, no cells. But again, we know how to get genomes. So we have no markers. We don't know. The only thing we know is this RNA polymerase. There is almost nothing else that is annotated. But again, that's why I wanted to mention NVO, because with the platform NVO, I can visualize all of it and gain confidence. It was very clear that those were genomes, about 300 kilobase. There is some variation. There is almost nothing known again, but it's something in terms of evolution that is very distant from anything else. So we got them. We got maybe 100 of them. And there was a lot of functions related to giant viruses that people were studying, but there was nothing related to making the particle. Again, at that point, I'm very new to viruses, so I don't know the problem, but I'm explaining that it is a problem. Because in order to place it in terms of taxonomy, it is based on the major capsid protein. So if you don't have it, you don't know what it is. So at some point, we released a preprint suggesting it is a new class of the classic giant viruses, and acknowledging that we did not find the key genes for making the particle. Next door of Patrick Forter and Morgan Gaia was Mark Krupovic, and we had a Zoom call with him, and we explained all the story. And we had core genes, so genes that are always present in all of those genomes, no annotation. We have maybe 30 of them. So if the major capsid protein is somewhere, it is in these core genes, okay? So Mark suggested to look at it, being an expert on finding major capsid proteins. And I think it took him less than a day to find it. And then we had another Zoom call, so maybe the day after, I'm not sure, maybe 2 days after, and I could see he was excited somehow. And he said, I found it, and it is not what we thought it was. It is not related to the classic giant viruses. It is related to herpes.
Okay. So
we brought him, happily so, and we updated the preprint. So it is also a good story of open science. So sure. A preprint is out, someone has additional knowledge, comes in, and then there is a publication. So that's how we found them. Then we got the major capsid protein. And we thought, maybe not all mirusviruses have the RNA polymerase, but they surely all have the major capsid protein. So we just came back again in looking at the metagenomic data of Tara, not from the eye of the RNA polymerase, but from the eye of the major capsid protein. And we got many more of them that were distantly related. So if you do a phylogeny of the major capsid protein, you will see one clade that we already found that contains the RNA polymerase, and many other clades that do not contain the RNA polymerase. Then more people came in, including Eugene Konin, and all of those people with knowledge and our work on including a lot of comparative genomics and so on, we realized, okay, there is this one group that has many functions related to being independent for possibly making a viral factory in the cytoplasm. So, it took me time, but it was to answer your question. And then there was other clades that not only did not have all of those functions, no single polymerase as far as we can see, which even herpes has a DNA polymerase. Yeah. On top of it, they are full of spliceosomal introns, which is something that you have to resolve inside the nucleus with a spliceosome in order to have a mature RNA sequence that will translate into protein. So it seems within the scope of this phylum, there is two states. One that seems to be able to replicate in the cytoplasm, and one that is completely stuck in the nucleus.
So among the mirusviruses, you get both. Is that what you're saying?
Yes.
Yeah. Okay. And so having an RNA polymerase is not a determinant because it could be cytoplasmic or nuclear. So herpes has an RNA polymerase, right?
No.
Does not? So maybe that's why it needs to go in the nucleus then.
Yeah, I'm not sure. It was part of the discussion this week. It's not clear if there is not one single gene that will tell you if it's stuck in the nucleus or the cytoplasm. In our case, the situation is quite clear because there is a series of functions related to multiple modules for transcription, replication, and making the nucleotides. On top of it, there is all of those spliceosomal introns that really have to be solved in the nucleus, and I didn't say it, it's important, they are enriched in the virion module genes. So the MCP is full of introns, the terminase is full of introns. Those are late genes. The late genes with introns tell you that the full cycle is in the nucleus.
You can use pox viruses as a model for it's cytoplasmic, right? You get all the genes, not just for the polymerases, but making nucleotides and so forth. That's what you need, right?
It's true. It's from another class of the other phylum. Yeah. I don't know, because of the divergence, how you can connect, if they have the same, I don't know.
Well,
but it's a good point.
Just by function, right? You know exactly what you need to be in the cytoplasm, completely independent of the nucleus, right? You need a lot more than the polymerases. And the pox can give you a list. It may not be definitive, but
it is a good point. It is a good point.
So the thing is, if you don't have an RNA polymerase, you have to go in the nucleus.
No. The story with the RNA, I emphasize the RNA polymerase because that was how we found Mirus. Okay, yeah. The story of the functions between cytoplasm and nucleus involved maybe 20 functions that include this one, but that's not the key one.
But I think if you don't have a RNA polymerase, you have to be in the nucleus. Right? I'm thinking about it because that's where transcription occurs in a cell. And if the virus doesn't have it, it needs to...
Can someone confirm that algae virales doesn't have RNA polymerase? Morgan?
Most of the algae...
Yeah, but are they able to go in the cytoplasm?
They are known to replicate in the cytoplasm.
Without the RNA polymerase. So it is possible without it to be in the cytoplasm.
I think so. So the virus genome that you found, does it have an RNA polymerase?
Yes, yeah. So Arantio, and we see, N of 1, but there are capsids obviously being made in the nucleus of the cells.
And are there many introns in your genome?
There are some. It turns out it's less the particular class of mirusvirus to which Arantio and friends belong are one of the ones that are relatively intron sparse, shall we say.
So from our perspective, so working independently with, so we created a gene model for trying to solve the gene calling of Mirus. we detect quite a bunch on your genomes, but then it's just, automatic bioinformatics to be confirmed. So possibly there is quite a bit.
Would you also agree that if you have introns that have to be removed, that has to happen in the nucleus?
For the spliceosomal introns, it is my understanding.
Yeah. Okay. Yeah. So that's another signature for nuclear presence.
It may be the biggest one. To have all of those spliceosomal introns in the late genes is quite a strong.
Okay. So let's just wrap this up by saying what's next with these. So in terms of mirusviruses, what do you want to do?
Yeah, I'm happy to start. We published it 2 weeks ago, and now it's vacation time.
Okay.
For a little bit, at least. We're so close to Christmas. But more seriously, well, there is a lot of unresolved questions. There is a lot of them. And I will try to stay close to Mark Krupovic and Eugene Kovnin, who wants to solve some of the evolutionary questions on if mirus emerge from a tailed phage. There is work to be done to try to understand when and who and clarify this aspect. The link to herpes also, there is a lot to be done in terms of taking advantage of 3D structures that we can now do at scale. So there is this aspect. And now we have a big expedition in Europe called TREC, which is to do similar to Tar Oceans, but on the coast line. TREC has multiple aspects of it, but one of it is aquatic. So we will explore mirusviruses in the coast, see if they are different and if they seem to infect different types of hosts.
Great. Okay, what about you, John?
Well, we've, in parallel with the dissection, I guess you could call it, of Arantio, the sort of cell biology, what's going on with this virus. So we have ongoing projects that are now trying to get a better sense of whether there are particular growth conditions where the episome versus the nuclear integrant are ramped up or ramped down. We don't really understand any of that, so there's probably years worth of detailed lab work to be done there. In parallel with that, we've got isolation of new Thrausto strains from nature and sequencing their genomes and asking questions, mirusvirus, yes or no? We have one no so far and a bunch of yeses. We're collaborating with Hiro Ogata in Japan and his team to really better understand the diversity within this particular lineage. So that's ongoing. And I think more generally, the question that interests me, and it's stemming in part from Tom's work and others, is the diversity of hosts. So there are these signatures from the genomes that tell us that, hey, lots of different eukaryotic lineages probably are harboring mirusvirus to some extent, and we don't have any definitive evidence of that. So there's a couple of groups that we're particularly interested in targeting. One is what are called cryptophyte algae. So these are photosynthetic microbes that we worked on for many years, going back to 2003. And as it turns out, science is funny that way. You loop back to some of the organisms you studied many years ago, and probably some of the ones we did genomes for back then are hosts of mirusvirus, but at the time we had no idea. So it's gone full circle.
So Tom, are we, in terms of the total number of different viruses on Earth, where are we on the curve?
On which one we identified? Is what you mean?
Is it saturated or not yet?
What we know, right?
Yes.
What we know.
What we know.
Oh.
Is it half, 10%?
No,
50%?
I am very confident with some data not out yet that it's not saturated. Now, is there a lot of stories to the scale of diversity of what we discussed tonight? I don't know. I hope so.
Yes.
I am very confident it is the case because there is a lot of ecosystems that have been understudied. From my perspective, my sense is what was sequenced. And there is a lot of ecosystems that were very poorly sequenced, and who knows what they hold. from my perspective.
So that's the question.
So it is not saturated.
Okay.
Of course not. I would like to just credit Ulysse Cuille, who is here doing a postdoc at Genoscope and who did a lot of work on especially identifying the most cryptic genomes of mirusviruses from all of those metagenomic data, and I did not mention him, and I should have.
Okay. Susana, what are you doing these days?
Well, I think we have a more organismal view of the story. So, because we have a lot of tools to work with the ectocarpus, so we're trying to understand how the virus or the provirus is silenced in these vegetative tissues and what triggers the activation of the virus in that particular cell type. And we are looking at, so we are using CRISPR-Cas to knock out several genes in the integrated viral element to understand the function of the different genes. And we are looking at defense systems because nothing is known in these organisms about how do they defend themselves against virus or bacteria.
That would be cool.
So, yeah.
All right, I have one more question for all of you. What do you do when you're not doing science?
Renovating a house.
Renovating
a barn, an old house in the middle of nowhere.
You should make YouTube videos. You can make money. What about you, John?
I ride my bike. I took 20 years off of any serious cycling, but got back into it back in 2012 or so, and so that's my addiction when I'm not working.
And so you take it wherever you go, your bike?
I don't take it everywhere I go. I wish I could. If I'm going somewhere long enough, then I certainly would, yeah, especially in wintertime.
So Harold Varmus is a big cyclist too. I don't know if you know the name.
I do.
But he used to bring his bike everywhere.
Right.
Yeah.
I did not bring it here.
Yeah,
I wish I had.
What about you? What do you do when you're not doing science?
Oh, I take care of my kids and my cat.
That'll do it.
And I play cello.
Cello. Cool. Why didn't you bring it here and play?
Yeah, I know.
Too big?
Next time.
You have to remember next time, right? Okay. That's TWiV 1281. You can find the show notes at microbe.tv/TWiV. Questions and comments, TWiV at microbe.tv. And if you enjoy these programs, we'd love your support. I know some of you are supporters. Go over to microbe.tv/contribute. I want to thank the meeting organizers, Matthias Fischer and Thomas Hackel, for having TWiV for the 4th time. I guess you like it.
Absolutely.
Good. So maybe a 5th time is in the future. That would be cool. My guest today, John Archibald, Dalhousie University. Thank you so much.
Thank you
for joining me. Susana Coelho is at the Max Planck Institute of Biology in Tübingen. Thank you so much. Yeah, it's good.
Thank you. That's good. Perfect spelling.
Tom Delmont is at Genoscope and Université Paris-Saclay. Thank you so much.
Thank you.
I'm Vincent Racaniello. You can find me at microbe.tv. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV. Ronald Jenkins for the music, and Jolene Ramsey for the timestamps. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.